Hey everybody, this is Dr. Duncan with BI 225. I'm going to be producing this video that's a summary of paper 4, which is Dowdy and Connor 2019 Nonchalant Flight in Tiger Moths is Correlated with Unpalatability. We're producing these videos to help out students since we're not having the class time discussions about these papers, and for students that can't join the video chats. Before we get started, and as a side note, it's fun to point out that both Dr. Van Zant and myself have been to the research station where this study was conducted. Dr. Van Zant visited there several years ago doing some research, and I actually spent a summer there as a volunteer when I was in college. They have a volunteer program where you work for 20 hours a week doing things like working in the kitchen and things like that. And then the other 20 hours of your work week, you get to tag along with scientists as they're doing their research. Or if you want, you can go hiking and exploring the mountains, which is pretty fun too. It's a great experience for anybody who's interested in doing field work as part of their career. You get exposure to a lot of different flavors of science and different types of scientists and all sorts of different personalities. It's a really good experience. Check in with me if you're interested in doing that. I can hook you up with some resources. Okay, whenever I read a paper and when I talk to students about how to approach papers, I always recommend that they do some things to kind of bring those papers to life. That's especially true if you're leading a journal club presentation about a paper, which many of you will be doing as part of your major or as part of your career. And I pulled up this picture which shows the moth diversity that you can get at in southeastern Arizona during the peak of summer. Most of those animals on the sheet that are moths. That's probably like 4 or 5, 6 hours of black lighting where you've got lights that attract moths at night and then they land on the sheet. Dr. Van Zant can tell you more about this. This is his jam for sure. Okay, let's get to the theoretical background for the paper. The background, of course, has to do with aposematism, warning signals. This, of course, includes bright coloration. We've talked about that in recent classes. And a lot of species that are that have this warning coloration, they exhibit chemical defenses or other defenses that basically back up their warning. In other words, if they're warning potential predators that they are unpalatable or dangerous, then they're making that this is the guarantee that they've got something to back up that promise. However, as you know, mimicry is involved. In Mullerian mimicry, you've got multiple species mimicking each other. These would be multiple species that are also unpalatable or a potential threat to predators. So Mullerian mimics are true mimics. They're not trying to deceive their predators. On the other hand, you've got Batesian predators. Those are ones that mimic the species that are toxic or dangerous. They are, if you want to think about it this way, they are cheating the system by looking like something that's dangerous without having to invest all the energy needed in order to create the defense structures or the defense chemicals that are needed to make them toxic. You got to keep in mind that these complex compounds that are used for chemical defenses take a lot of metabolic energy to create, and not just to assemble the molecules, but the whole process of obtaining them usually from their food and manufacturing them, and then not manufacturing them, but then sequestering them in their bodies in the places where they need to be. That's a lot of energy devoted to defense. If you can avoid that and still get the same benefits by looking like something that's toxic or dangerous, then that can give you a big advantage. I want to just take a sidebar here to talk a little bit about the history behind all this, which I don't think we really covered too much earlier. This guy Bates, who's depicted in the picture at the right, he was what I would call a biological badass. You might have heard of Alfred Russel Wallace. Wallace was the co-author with at the presentation of the idea of natural selection. The co-author with Wallace, of course, was Charles Darwin. Darwin had come up with the idea over a decade before, but sat on the idea and didn't share it with other scientists. Wallace also came up with the idea of evolution via natural selection, wrote to Darwin, and the two of them co-presented to the Royal Society. And Bates factors into the story because Bates and Wallace were partners in collecting organisms from around the world and then selling them to wealthy collectors. And that's what exposed both Bates and Wallace to a tremendous diversity of species. And there's a story that might be fun for you guys to look up sometime about how Wallace and Bates, after kind of getting bored with collecting butterflies and things like that in England, decided to head to Brazil, and they spent several years in Brazil, had many near-death experiences, and just had incredible adventures, finding some species for the first time, encountering some peoples that hadn't seen white people from Europe before, and these sorts of things. It might be worth looking into if you like adventure stories like that. Bates wrote about that in a book. Okay, back onto our topic at hand. One of the things that's seen with animals that exhibit aposematic coloration or other traits, they often show what's called sluggish behavior. So in other words, if you were to approach these animals, instead of them fleeing, like, they will often sort of either stand their ground or just sort of move away kind of slowly. And there's three hypotheses as to why this is the case. One hypothesis is that they are avoiding triggering an attack. Some predators will have an attack triggered simply by them seeing movement. I think of the house cat that I have lurking around here somewhere that sees something moving and it's on it. It acts first and asks questions later. And of course, that could be detrimental to any organism. And if you can avoid triggering that attack, then you avoid getting damage from a potential predator. A second hypothesis is that the slow movement itself is a aposematic signal. In other words, potential predators see an animal that doesn't look worried and is moving slowly. And I'm using teleologies here, of course. The predator is thinking to themselves, maybe that potential prey item knows something that I don't. Maybe there's something there I should be concerned about, and maybe I should keep moving and find a meal elsewhere. So that's the second hypothesis. The third hypothesis is that the sluggishness, the slow movement, makes sure that the predators have a chance to see this signal. So if you're a brightly colored animal and you're moving away very quickly, a predator might not have a chance to see that you've got the warning coloration, and they might attack. So these are those three hypotheses about why aposematic animals are often exhibiting sluggish behavior. And just to be clear, our authors of the paper refer to the flight behavior of moths as being nonchalant, which is just a fancier way of saying sluggish, if you will. All right, so let's keep moving here. So this brings up the question about whether or not animals should just flee anyway, even if they're aposematic. Right? Because if a potential predator is approaching, maybe that predator's never encountered this, the warning coloration that you have, and doesn't realize that you're warning them that you're potentially dangerous or toxic. Well, so shouldn't that potential prey item always flee anyway? Well, maybe not. There's some cost to this. There's the lost energy of fleeing. There's the lost opportunity. For example, you might be feeding or you might be hunting your own prey, and if you have to flee the scene because a potential predator comes along, you're losing opportunity. And also sometimes animals wind up falling prey to a different predator when they're escaping a first predator. I see this all the time when I'm out in the woods and I'm watching birds. There'll be one bird will be chasing another, like a hawk will be chasing a smaller bird, and then that smaller bird winds up in the talons of a different hawk that it never saw coming. So, prey are in general expected to balance the costs of fleeing or flight with that predation risk. One way to think about this is in terms of flight initiation distance. If you're a prey item and every time you catch even the slightest glimpse of a predator, no matter how far away they are, and yet you flee the scene, you're going to be spending a lot of time in flight mode instead of feeding or doing some of the other activities that you need to be doing. But if you always wait to, well, not always, if you wait until the last minute, maybe you won't be able to escape. So flight initiation distance is one way of measuring like where those curves cross, where the risk, the predation risk curve crosses with the benefits of not fleeing. So for folks that have studied aposematic coloration, they've considered this in terms of how flight and initiation distance might be influenced by chemical defense. And this potentially explains the sluggish behavior. So animals that are sending bright, bold signals that they are toxic or unpalatable or dangerous to potential predators, that their flight initiation distance is somewhat shorter. They will let predators get potentially a lot closer. So this is some of the theoretical background that is behind the study that we're studying now. The authors actually summarize this rather well, and I pulled out this quote here. This is from the paper. They say that chemical defenses may be one such factor influencing flight initiation distance. However, the escape responses of chemically defended prey have not been well studied, and it is not clear whether sluggish animals should be expected to remain so when directly threatened. This is what I would say is what's the gap in knowledge that they're trying to fill. Every research project is trying to fill some gap in knowledge. Identifying the gap is really important for understanding the motivations behind why a study is done. Okay, let's talk a little bit, excuse me, let's talk a little bit about the mechanisms that are involved, the species that are involved here in this study. Some of you probably already know this, but I thought it'd be good to review anyway. Bats, particularly microbats, that's opposed to megabats. Megabats are largely fruit-eating bats that are much larger and they don't do sonar. But microbats use sonar to detect their prey or their food items at night in the dark. Some microbats eat insects. Many also in the tropics eat fruit, and a few do things like catch fish out of ponds, and some actually pollinate flowers and things like that. But the ones that we're focused on are insectivorous, and they're using their sonar to find prey items that are flying around at night. So if you don't know what sonar is, it's a method of sending out high frequency sound signals and then detecting the signals as they bounce off an object and reflect back to you. And so this is something that's used by bats, as illustrated in this cartoon. It's also used by cetaceans to detect things underwater in the dark or gloom of the ocean. And this is also something that humans have done as well. We use sonar. It was initially developed I think it was initially developed by the military in order to aid things like submarines and so forth. Okay, so that's how sonar works. Let's move on now to the moths that are involved. in the study. The tiger moths, like many moths and butterflies, as larvae, they are eating plants that themselves are producing toxins. The many species of lepidopterans, which include the butterflies and moths, will sequester these toxins in their body tissues as a way of chemically defending themselves. A famous example, of course, is the monarch butterfly. Many of these species that do this exhibit their own aposematic coloration in order to warn potential predators, hey, don't mess with me, I taste nasty, you don't want to waste your time, and frankly, I don't want to be chewed on a little bit while you figure that out, so please just stay away. Again, apologize for all the teleologies. All right, so this is one of the defenses that tiger moths do. But tiger moths also have another technique that's thought to be a part of aposematic anti-predator deterrence, and that is that they make noises. This might be surprising to you to know that that moths can make noises. It is true. And the noises that they make have been hypothesized to be jamming signals that interfere with the sonar that's being used by bats. It's also hypothesized that the anti-bat capture noise is its own form of aposematic signaling, where basically potentially toxic prey items, the moths, are sending an acoustic signal to the bats saying, hey, don't bother trying to capture me. I taste like crap. Just keep moving. Go somewhere else. I put a link in here. Oh, by the way, those are not mutually exclusive. Those could both be those could both be favored by natural selection at the same time. Those could both work simultaneously. I put a link in here if you want to see some video of moths using this signaling at the point where bats are about to capture them. It's pretty cool, and you can see this in action. This will be in the PowerPoint slides that are posted on Moodle associated with this. Now, interestingly, with regard to the tiger moths, that many of them have what's been described as nonchalant flight or sluggish flight. In other words, they're in a hot zone where there's bats feeding, and they just sort of casually fly through the night as if they don't care about the bats whatsoever. And of course, that raises all sorts of questions, which our authors are trying to answer here today. It's also been noted that as part of that sluggish behavior, that some of these moths will try to evade bats when bats get close, whereas others don't. Of course, the main form of evasion is to go into a steep nosedive and almost as if they're dropping to the ground which is the fastest way to get out of the kill zone where the bat's coming into. All right. Here we're looking at a critical paragraph in the background section of the paper. I would say these authors do a fantastic job of really clearly highlighting what their purpose is and their hypothesis is. So let's look at this first part of the highlight up top. This is, and I'll just read this out loud. This is where they nail down this is what I call their statement of purpose, or SOP. In this study, we examine the role that unpalatability plays in predicting the likelihood that individuals of a given species will enact escape behaviors in response to the threat of predation. So basically, they fold in all that theoretical background with the gap in knowledge to say what they are planning on doing. That's their statement of purpose. When you write a good paper, a research paper, a lab report, you need to make sure you have something that is equally clear and deliberate. Okay, they quickly go on to say what their hypothesis is, which is now fully motivated by the statement of purpose. They say, we hypothesize that more unpalatable tiger moth species would be less likely to exhibit escape maneuvers than their unpalatable counterparts. And that reminds me that I didn't say earlier that tiger moths vary in their palatability. Some had already been known to be pretty toxic to potential predators, whereas others were already known to be less toxic. Okay. The other thing that I'd point out here in this paragraph is that the authors nicely frame the importance of their study to their field of science. They say this at the bottom. They say this is the first study to comparatively examine the escape behaviors of aposematic insects in a natural context. Compare the likelihood of escape between both aposematic and mimetic species, and construct a predictive model of escape behavior. Okay, so basically they're highlighting and rightfully bragging about three major contributions to their field right there. Okay, that wraps up the background section to the paper. The next video will cover the methods.